NCIS, CSI, Bones, and Forensic Files. We have all heard of these shows, and since the early 2000s, these types of shows have dominated the entertainment world. Unfortunately, this has led to an unrealistic expectation in the field of forensic science. But what is forensic science? It is the application of science to the law and investigations, and there are many subdisciplines. In this video, we will focus on forensic DNA. The era of forensic DNA began with Dr. Alex Jeffrey's discovery of DNA fingerprinting in 1984. In the following decades, research focused on exploring DNA profiling. With Jeffrey's discovery, genetic evidence could now provide the ability to discriminate between two individuals. Therefore, it is important to maintain the integrity of DNA because it can be a vital piece of information in a court of law. Contamination can occur at any point in the investigation process, and there is extensive research understanding the potential for contamination. Thanks to next-generation multiplex, the sensitivity of the technology's analysis has increased the accuracy of the contamination rate. Despite the research indicating the problems contamination causes in forensics, the contamination rates have been increasing, with the highest percentage of contamination originating at the crime scene during the collection process. At a crime scene, contamination occurs through either primary or secondary transfer. Primary transfer happens when an object comes in direct contact with an individual, while secondary transfer is a result from indirect contact with an object such as a sneeze or uncleaned equipment. Why is this important? The CSI effect is an increasing phenomenon that occurs because jurors have an unrealistic expectation of evidence due to television and expect DNA evidence to be entirely foolproof. Because of these expectations, genetic evidence is often weighed more in the decision of the verdict. Therefore, it is even more vital to maintain the integrity of the DNA in question. Contamination interferes with the analysis of DNA profiles. People have still been wrongly convicted based on genetic evidence because contamination is not always easily detected. In Australia, DNA analysis connected a boy to the crime scene and was then convicted of the crime. However, the DNA had unknowingly been contaminated during the collection process. In Switzerland, a study found 227 of the detected contaminations involving investigators likely occurred at the crime scene. One of these contaminations was caused by the crime scene photographer, and another 23 of the contaminants was from individuals who were simply present at the crime scene. In Austria, a study found between 2000 and 2016 that 67.1% of the contaminations originated from crime scene investigators. During this time, swabs made up 45.8% of the contaminations from the collection methods. In the Netherlands, between 2008 and 2012, contaminations went from 49 to 135 at the Netherlands Forensic Institute. With better training and a broader awareness of how contamination occurs, then the contamination rates could be reduced, and therefore preserving DNA integrity.